From Microbe TV, this is Office Hours for Wednesday, the 25th of September, 2024. I'm Vincent Racaniello, and welcome to your viral corner of the internet. We're here to talk about viruses, and you're here to talk about whatever else you want to as well. Tell us where you're from. We'll give you a shout out. But meanwhile, let's thank our moderators for coming tonight. I see in the house we have Les. We have Frank from New York. And we have Vanity Nutrition from New York and Tom from the Oregon Coast Range. Thank you all for joining us and keeping it civil. And thanks everyone for joining us tonight. This is my second live stream of the day. Not complaining, just like the opportunity to be able to interface with all of you. I heard a great quote today. Let me actually type it. It's such a good quote, and we have to remember it. The quote was, "If you do your best, sometimes you will resonate with someone." Okay? There you go. I heard this just an hour ago. If you do your best, sometimes you will resonate with someone. So the reason I like this is because I've always tried to do my best. Not try, do, right? Do. Never mind try, just do. And the result is this community of people who for whom the things I do resonate. Let's put it that way. My teachings resonate with you. And so I love this statement because I'm trying to do my best, and sometimes I resonate with many of you. I was looking to speak of resonate. I was looking at the most popular, the most viewed videos on the YouTube channel. I was curious because I want to try and reach more people, right? The most viewed video is "What is a Virus for 2020?" It was released to just the early year of the pandemic. 675,000 views. So people sought out virologists to understand what viruses were. So that was great. But unfortunately, as the pandemic went on, they sought out charlatans who presented themselves as experts and whom we fought for months and months and months here trying to dispel the things that they said. So let's put up this quote at the top of the page for today. We'll keep it there. By the way, if you enjoy these conversations about science, not just this live stream, but also all the podcasts, This Week in Virology, etc., please, we'd love your support. You can see up here. Nope, that's the other finger. Venmo. We have a Venmo, and we have a webpage, microbe.tv/contribute, where you can find several ways to donate. And if you are in the U.S., your donations would be federal U.S. tax deductible because Microbe TV is a nonprofit entity. It's a nonprofit where, in theory, our revenues go towards programmatic functions, as they say. Now, tonight, as always, I want to answer your questions, but I'm also going to talk again about polio because I think this is really important. And I'll explain in a moment. But let's see where people are from. Let's see where people are from. John. John is from the Twin Cities, Minneapolis, 74F. What's the weather like here? Let's take a look. It's getting fall, 18C here in New Jersey, and the humidity is, well, it's 85%. That's pretty high. Welcome, John. Hello, Questella. Questella was at our Twivo live stream today. Welcome. Questella is from Ohio. I know this. Artemis is from Montreal. 16C and light rain. Laura. Or is it Laura, Kip and Laura are in San Francisco. Welcome. And thank you, Laura, for your support of science communication. It's our little grassroots effort. We do our best, and sometimes we resonate with someone. And in San Francisco, it's 65F. I have 82 on Monday. Cool, cool, cool. That's where you're from, folks. Wayne is from Urbana, Illinois, mostly sunny, 69F. Kathleen is in South New Jersey. Lise is in Columbus, Ohio. Got your Novavax. Now, we have Tatiana Beast Mode, very active in the chat. But where are you, Tatiana Beast Mode? If you'd like, you don't have to tell us. Frank is in Santa Barbara, 72F. Okay, Tatiana's from Birmingham, Alabama, 77F25C. Thank you for telling us. Apparently you have a hurricane on the way, eh? Okay, good luck with that. Kang is in Chicago, 20C and sunny. Betty is in Washington Heights, New York City, my old stomping ground. Well, technically still, right? I'm still a professor at Columbia. I teach a very popular virology course in the spring. But I don't go to Washington Heights to do that. So Washington Heights, of course, is where the medical school campus is. And my office was there for many years in my lab at the Hammer Building, which is at the intersection of Fort Washington and 168th Street. And I closed my lab in 2022, and they promptly took my office away. So I have no digs up there. I go to the incubator or Morningside Heights when I teach. There you go. Hans is in Guayaquil, Ecuador. Welcome, Hans. Cheryl is in Santa Rosa, 67F and sunny. MN is outside DC. Zambre is in eastern Pennsylvania. Yeah, we saw you on the Tweeva live stream earlier today, for sure. I remember eastern Pennsylvania. It's kind of a, yeah, people don't say eastern Pennsylvania, I guess, but it struck. It reminds me. I don't know. Will, hello, Changsha, China, 23C and hazy sunshine. Pete is in London, remembering his mother who would have been 101. Excellent. Michael is in Mexico City, 18C. I'm resonating with thousands and thousands. Well, I hope so. That's the goal, right? And that quote resonated with me. Actually, it's funny that it's resonate because I was watching a video of a fellow in Japan making handmade guitar. And he said that at the end. He said, if you do your best, sometimes you resonate with someone. And it's funny, the guitar is resonating, right? But in a broader sense, it's a great quote. Gallbladder, thank you very much. Do or do not. There is no try. Yeah, I don't think it's useful when you say to someone here, do this, and they say, I'll try. It's almost like setting themselves up for failure. You know, I understand people have limitations. We all do. But shouldn't you say, yeah, I'll do it? And then if you can't do it, you come back, you say, here's what I did. And couldn't get it to work. But don't say I'll try to begin with. I feel that that's uh, underachieving, right? Something like that. What is it? It's setting a low bar for yourself. You shouldn't do that. Brian is in Sydney, Australia, rainy and 10 degrees Celsius. Ooh, Sharon is in Tasmania. How cool. Lots of people on that lower part of the world tonight joining us. Mike and Lori, Seattle, 60F. Spartavus, Weymouth, UK. You adore our vids. Thank you. I'm glad you like them. Adoration is a very high standard. You know, I would never say I'm going to make videos that people adore. And so, therefore, when people adore them, wow, that's cool. But I don't say I'm going to try and make that video. I say, yeah, I'm going to make it. Got to be positive, right? Doreen. Hello, Doreen from Elgin, Illinois. Lisa is in Sperryville, Washington. Resonate all the way to Tasmania. That's great. I had a feeling that you folks here would appreciate the resonating, because I think you, all 100 of you so far, what we do at Microbe TV resonates with you. That's why you're here. So I can skip a week and you're back because there's something that resonates with you. And it's probably a number of things. First of all, it's information, right, from a good source. But it's also the fact that I'm honest, right, with the information. I don't do things for the sake of publicity or money. I try to tell you like it is. And I think that kind of honesty is appreciated. And also expertise, right? Although many people don't care about that, but the people here do. Yeah. Hello, Nebby is in Florida. G just watched 2 of the most popular videos. Yeah. You know, it's interesting that the most popular one is only 675,000 views. Now, there are many channels where a video will go up and within hours it will have 2 to 3 million views, right? I don't think that's unachievable, but we haven't reached the formula yet. I'm not going to say I'm going to try and do that. I'm going to do that. That's a goal, is to achieve a few million views per video. But it's some kind of magic that we haven't identified yet. I'm very much in awe of people who can get 2 to 3 million views. I mean, for non-controversial things. I mean, if you take your clothes off, sorry, that's too easy to get 2 to 3 million views. But if you're talking, like Marcus Brownlee is an example. He's a tech reviewer, and he sits in front of the camera and talks pretty much and gets millions of views. So a lot of people want to know what he has to say. That's great. So he built a following based on his opinion, but he's just talking. He's not jumping through hoops or anything like that. And I appreciate that. I mean, the videos are well produced, but it's really him in the end talking. That's very impressive. Angela's in Manhattan. Click the thumbs up, folks. Yeah, we got 108 people. And it is, we have 64 likes. Hit the like button. It's down below the video because maybe somebody else will come over in the middle of this live stream and learn something. Yes, yes. To have 1150, Tom writes, letter from Florida with respect to the mRNA vaccine. Daniel and Vincent were restrained and polite in their response to dangerous nonsense. I think it helps to be restrained and polite, even though sometimes you'd like to say, what? And go off the end. And, you know, sometimes I do. I get annoyed. But I think restraint is the way to deal with most issues. Because if you stay calm, people may feel that you have they have more respect for you, maybe. The contribute links are covered by the chat. Yeah, I see. Yeah, they got that. Well, yeah, the Venmo one is okay, and the microbetv.com/contribute. You know, YouTube puts up all kinds of stuff all the time. My audio is pretty quiet. Yeah, I can crank up my audio. Check chat. Hello, 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 hello. You know, all I have is this. Well, that's my headphones. Let's. It doesn't help to put my headphones up. Let's increase my microphone volume. Yeah, now it's peeking into the yellow. So let me know if that's better. Will. I know a lot of people who had their vaccines who are now skeptics. Yeah, I don't understand. You had your vaccine and it probably kept you out of the hospital. Why are you a skeptic? Because you're listening to someone who doesn't know what they're talking about? Here's the thing, folks. If you want to make an opinion, you need to search out the right information. And to do that, you could do the literature, right? That's not hard. I'm sorry, that's not easy to find the right paper that addresses what you want to do. You could listen to someone, but there's so many pundits out there who want to tell you what they think to get you to be on their channel. You know, they want your hit, and they're not necessarily telling you the right thing. And that's happened throughout the pandemic. So it's not surprising that people turn skeptics. Oh, I Googled this and this is what it said, so I'm not taking that vaccine. No, you're wrong. Sorry. Jill is from Wilmington, Delaware. Volume's good now. Very good. Thank you very much. If there's ever a technical issue, just tell me. I will fix it. I won't try. I will fix it. Here's a story. So in the last clinical update, 1150, if you went to the web page, it said clinical update, TWIV 1150, and it said October something 2023. And Kim, our social media person, she texted me, something wrong here. And I looked at it and I went to the web page and the date was right. And I said, this is above, this is like a glitch. So I said, is there a WordPress update? And there was. I did it and I applied it and it fixed it. So when in doubt, 1, make sure it's plugged in. 2, reboot. 3, update the software. Of course, updating may break it. For example, a few weeks ago, I bought a teleprompter, which is a device that sits in front of the camera, and I can look at the screen and be looking at the camera at the same time. And so I would put, the live stream chat up there, and I could read it, and it looks like I'm looking right at you. A software update with the Mac broke it. Doesn't work anymore, so it's off. Now I have to look at the lower screen, and you can see my eyes are not directly looking at you, which in the big scheme of things, doesn't really matter. But, over the years, I hone things to make them work really well. And I think if you're looking at people, that really is effective. So software updates can break things as well. That's the point. Yeah, there's another paper on the market origins of SARS-CoV-2 just come out. We're going to cover it on Friday's TWIV. And so that is more evidence that, animals susceptible to SARS-CoV-2 were present in a part of the market where there were also SARS-CoV-2 sequences found of both lineages. So basically, evidence continues to accrue to support a market origin of the virus. And no evidence for any other scenario accrues whatsoever. So you must question why people have another idea for which there's no support. You can have ideas, but don't say this is what happened, right? Which is what many people do. Sherry is in the north of England. Elizabeth is in Mississauga, Ontario, where it's 20°C. Elizabeth is in West Virginia. Brandon is in Ann Arbor, 67F. John says someone on social media said viruses were interesting, but she was struggling in class. I pointed her to Vincent's free lectures and she was thankful. That's what I call resonating with someone new. Totally. You do your best. I work really hard at my lectures over the years. I've got them working really well. You put them out there year after year. You update them. You make them available to everyone. That's the key. You do not charge for them. And I have to say to Columbia, thank you for allowing me to put the lectures online and not worrying, because your classrooms are full. It doesn't matter if my lectures are available on YouTube, right? So thanks, John, for pointing that out. That's really great. Hat is in the San Fernando Valley. We're at 76F. Yuxing is in Hangzhou, 24C. It's morning there. Dean is in Wisconsin. Stark is in Nelson, BC, 28C. Hello, Carol, Dr. Nephrologist. Bakersfield, CA, 96. Wow. Wow. Very hot. Yeah, I haven't frozen yet here. I still have a little bit of summer warmth left, but I can imagine in December, January, one would like to absorb some California warmth. All right, so Sharon has organic magnesium blend in almond milk instead of tea. This is very interesting. Andrew. I don't know if I said Andrew was here, but there he is from New Zealand. I've added some lighting in the background. Yeah, I did. So Kathy Spindler said my background was too dark. So there is a tube light leaning behind this bookshelf here, which is spilling colored light onto the area. And it can be changed to different colors. And then there is, you can't see it because it's out of focus. And so actually there is a virus particle there, an icosahedral particle with a light shining down on it. I also blurred the background a bit to separate it from me. How did I do that? I got a lens that could open up wider and have a shallow depth of field. These are little things that I like to tweak to improve the experience. We'll try not to try. No try. Just do. Mark, thank you for your support for science communication. My mom is 89. Is it worth getting her an RSV vaccine? Well, this has been asked of Daniel multiple times. And so I will channel his answer because, as you know, I'm not a physician and I should not be giving you medical advice. So Daniel says yes, because nursing homes are scenarios where you can have spread of RSV infection because many people in a closed quarter, a lot of contact between them in rooms, and they're in rooms playing cards, right? And then the caregivers come and go, as well as other people, the family, and they bring viruses in and inoculate the facility. So yes, it's highly effective, over 90% effective at preventing severe disease. Durability, of course, we don't know yet because it hasn't been out that long. But I think Daniel has said many times this is a good candidate for this vaccine. Let's see who else is here. Condor Kid is from the UK. Ooh, do reaction videos to science content. I really like to do this. I was watching a sci-fi movie the other day, or TV show, and the lady was talking about making a recombinant protein, and she used like 6 words that were so complicated, all buzzwords, and I felt like I should just play that and make a 30-second reaction, right? What? I'd love to do that. I would really love to do that. So yes, Zombie, I'm going to do that more often. Oh, yes. Honesty goes a long way. Admitting you don't know something. I learned a long time ago. First, if you don't know something, admit it. And people like that. And second, if you're wrong, admit it after the fact. And I've done that multiple times, and that really, really resonates. I say, oh, I think this is the way it is. Nope, I changed my mind. I'm wrong. I saw other facts that mean I'm wrong. And you admit it. Really hard for people to do. I don't have a problem with that. Why? I mean, why do I have to deny things? No, I've had a great career in viruses. I built this science communication company. I built a community of people with whom we resonate. Why would I have to fake anything? I feel like I've accomplished good things. Not to be immodest, but just why would I not admit that I don't know something? And so if you ask me something here, I'll say I don't know, or I'll look it up. So sometimes I can look it up and find it really quickly, and I can tell if what I've looked up is correct. Yeah. So Spartacus said, I think you nailed on the head regarding resonating and honesty. This is exactly why I tuned in 4 years ago to learn the truth. Thanks for all that you do on your channel. You're welcome. I haven't done anything different from what I did all my years as a professor, right? Be honest with people, tell them like it is. Tell them when you don't know something and admit when you're wrong. And I brought that over here and it worked. And I didn't have to make a persona, right? So thank you. What's your new streaming system? It's the same streaming software, Ecamm Live, except the blue thing is gone. Where did it go? Now, every time I touch something, there's supposed to be a blue frame here. Where did it go? Let's see if I can get it back. So here's the thing I use. Yeah, so you see, that's when I have a guest, that's what it's supposed to look like. So let's see, show in all, let's put the rectangle in all the things. And so let's see if it's back. And now it's covering me. So this is the problem with doing something, doing something live. I put this rectangle. It's at the bottom of the stack of stuff. I don't know why. It's weird. Well, we're going to have to not have the blue tonight because I can't figure it out. But normally I have this nice blue. I took it away from there and then moved it here, but it didn't seem to work out. But let's see for Dixon, a nice blue thing. Where does the blue thing reside? Show in the current scene all the way at the bottom. So VR in frame, show in the current scene. Well, let's try putting it all the way at the bottom. Let's see if that works. Hey, there you go. live troubleshooting, which you shouldn't be afraid to do. It's fine. Anyway, this is just a look that I built from the existing software. I have a blue gradient background and then some boxes with information in it. It's the same software. Okay, where else are people from? Oh, from Singapore. All right, that's far away too. Volume is good now. Let's see what's yeah, step 0, read the manual. Read the effing manual. Anybody else? There are lots of good questions here. I will get to them. Paso Robles, SRR, California. Margot is in Phoenix, where it's, wow, 108. Oh my gosh. Thank you, SRR, for your support of science communication. We really appreciate it. We run Microbe TV on your donations. We don't do ads. We want you to focus on what we're saying. Galloping is in Eugene, Oregon. Ito E is from Cincinnati. Yeah. I still have the recording. Yeah, this would occasionally play upstairs, and one night people loved that the ambiance caused by that. Yeah, I do think. Yeah, I don't have the sound here anymore, but I did put it down here. I could play it sometime. Let's see. Anybody else? Reaction videos, yes, but stay calm. Yeah, stay calm. Just get funny, right? This is interesting. This is a preview of today's mini lecture. What is your opinion of the situation in Gaza with respect to the polio vaccination effort? Well, I have a very distinct opinion, and that's one of the topics of today's mini lecture. So hold that thought. I will come back to it. Hello, Rima from Iowa. Welcome. And Jefferson Spaces in Miami. Well, everybody, welcome to the show. Not really a show, but welcome to class. Welcome to office hours. And you have a lot of questions, which are good, and I will answer them. But I want to talk a little bit about polio. And as you heard Mike ask, what's my opinion of the polio vaccination effort in Gaza? And so I want to talk about this. And I have talked about polio recently. I know that. I think it's an important situation. So globally, WHO and many other health agencies are attempting to eradicate polio. That's an amorphous term because polio can mean poliovirus or poliomyelitis, the disease. There's zero chance they will eradicate poliovirus. And I've talked about that before on this program. But they're trying to eliminate poliomyelitis. However, they've been trying since 1988. They haven't got there, and there's no signs of them getting there. Now, they have eliminated a lot of polio, but they've also caused a lot of polio. And so that's why the name of today's mini lecture is Man-Made Poliovirus Outbreaks. Because yes, most of polio, myelitis, for the past 6-8 years has been caused by humans, caused by the polio vaccine. Thousands and thousands of cases. And the more we immunized with that oral polio vaccine, the more cases of polio we cause. And that is what's been going on in Gaza. Now, I wondered about talking about this because I suspect that most of the U.S. and other countries where people are listening don't really think about polio much, right? They think it's not an issue. We haven't had a case. We had one case in 2022 in the U.S. of poliomyelitis. Most people probably think it's done and gone, but it's not. And it's getting worse. And so I'm talking about it a lot. I put 2 short videos up on the YouTube channel in the past few weeks, which express my frustration with how poorly things are going. And tonight I want to talk a little bit more about it. Okay, so man-made poliovirus outbreaks. So what is this about? So there's a website called Global Polio, here it is, polioeradication.org. And it is a website of the Polio Eradication Initiative, an organization whose job is in part to publicize the eradication efforts. And so this was an article published on September 13th. Around 560,000 children vaccinated in the first round polio campaign in Gaza. So as you know, there was a case of poliovirus was circulating in Gaza because of the poor sanitary conditions. It was a case of poliomyelitis. And so WHO coordinated a vaccination campaign, which has hit over 560,000 children. And what they have given them is OPV, oral polio vaccine, and specifically NOPV2. So as you know, there are broadly two kinds of polio vaccine. There is the inactivated polio vaccine, IPV, developed originally by Jonas Salk, which is used in many countries, including the US. It's injected. It does not cause polio. It prevents poliomyelitis. It does not prevent the virus from infecting you and replicating and shedding and spreading around the environment, but you don't have any symptoms. It's an asymptomatic infection. The WHO is not using IPV for its eradication effort because they claim it's injected, it's too expensive, and it doesn't work as well. They claim that OPV is the vaccine of choice. Well, you know what? They're wrong. As I'm going to show you today, OPV is not the vaccine of choice. Now, to provide a little further background for this story, and again, I discussed this a couple of weeks ago on office hours. In 2016, type 2 poliovirus was declared eradicated. There are 3 types, 1, 2, and 3. They all cause poliomyelitis. The vaccines are meant to target all 3. And in 2016, WHO said, we don't find any more wild type 2 poliovirus cases, poliomyelitis. So they said the vaccine, the type 2 component, actually causes a lot of polio. So we're going to remove the type 2 component from OPV. And then, since there's no wild polio, it shouldn't be a problem. It shouldn't be a problem. Why did they think this? Because they put numbers into computers and did modeling, and the computer said it would be okay. Well, they were wrong. It was the biggest mistake ever. The withdrawal of OPV2 from OPV in 2016 has led to thousands and thousands of cases of polio caused by the vaccine. And so here, what they do now is if there is an outbreak of type 2 polio, which is what that one child had in Gaza, and there's more circulating there, they go in with NOPV2, which is a single serotype, and NOPV2 was modified genetically to be, quote, better, unquote, than OPV2. But as I'll show you today, it's not. And they introduce more OPV2 into the environment. So that's what's been done in Gaza. So at the end of this article, this is the part I want to focus on. They write, novel oral polio vaccine is a polio vaccine being used to stop transmission of variant poliovirus type 2, currently the most prevalent form of the variant poliovirus. Now, first of all, what the heck are you talking about variant? Nobody uses variant to describe any poliovirus. A prevalent form of the variant polio. This makes no sense whatsoever. What is correct is that, yeah, polio vaccine-derived polio type 2, which is polio type 2, let's just call it that. It's causing more polio than anything else in the world. So NOPV2 was developed in an attempt to prevent OPV2 from causing polio. Well, it didn't. And I'm going to get to that in a moment. So it's a failure. But they say here, NOPV is safe and effective. Nope. Not safe. No more effective than OPV2. Offers protection against paralysis. Okay. Community transmission? Nope. No evidence for that whatsoever. It is the vaccine globally recommended for variant type 2 polio outbreaks. Now, this use of variant is wrong. It's poliovirus. It's not a variant. And it is not globally recommended. I will show you an article which says it's not. So why is this polio global eradication initiative writing this? Who knows? Someone was tasked with the job of making a press release. And they were given some information which is wrong because they're not going to go check the literature. So that whole paragraph, that whole last paragraph is wrong. NOPV2 is not a variant, it's not safe, it's not effective, and it doesn't block transmission. Yet this is what they are using in Gaza. In my opinion, they should have gone in with IPV and done the right thing. Now, with 560,000 doses administered, we're not going to see any paralysis yet. You'd have to get up into the millions. But that virus is now circulating in Gaza. That vaccine virus is circulating, and it will find kids who are not immunized, and they will get paralyzed by it. Okay, so this is the thing. This is what I start with. This article, I read it the other day, And this is my reaction, okay, here on Office Hours, a little bit extended. So, first piece of data. This is a Lancet article. Effectiveness of poliovirus vaccines against circulating vaccine-derived type 2 poliomyelitis in Nigeria between 2017 and 2022, a case control study. So this is a paper, a research paper, where they asked, how does IPV, OPV2, and NOPV2 compare in the ability to prevent paralysis? Just paralysis. So what they did is they looked at surveillance data from Nigeria. AFP is acute flaccid paralysis from 2017 to 2022. So they say, okay, we identify all the cases of poliomyelitis, acute flaccid paralysis, and we ask if they've been vaccinated or not, and if so, with which vaccine. And then they calculate effectiveness of a single dose of each vaccine, one dose only, against paralysis. Look at IPV, 89% effective against paralysis. That is a good vaccine. That's what we use here in the U.S. Monovalent OPV2, 1%. New novel OPV2, the one engineered from OPV2 to be safer, minus 23% effective. And basically, it's the same as MOPV2. It's not really good. One dose is not really good. IPV is much better. How can you say that NOPV2 is the vaccine of choice globally? You can't, because you haven't read the literature. That's why you can say that. And this is what the problem is. The people who write these press releases for GPEI and, the Gates Foundation, who funds a lot of this eradication work, and the WHO, they never look at the literature and ask, am I writing what makes sense? Nope, because what they write doesn't make sense. So here's the summary of the Lancet article. Interpretation. We found no significant difference in estimated effectiveness of the two oral vaccines, supporting the recommendation that the more genetically stable NOPV2 should be preferred in type 2 outbreak response. So basically they're saying it's no worse. NOPV2 is no worse than MOPV2, so we should use it. But they say it's more genetically stable, marginally so. I will show you the data for that in a moment. Our findings highlight the role of IPV in the necessity of strengthening routine immunization, the primary route through which IPV is delivered. Okay, what does that mean? So in Nigeria, kids get IPV along with a bunch of other vaccines in a pentavalent or hexavalent formulation, right? DTaP and IPV and others combined. It's not used for outbreak control. Yet it's really good at preventing paralysis. And if you use it, you don't seed the environment with infectious poliovirus. So this idea that OPV is preferred is just wrong. It hasn't worked for many, many years now. For 40 years of the eradication campaign, it has not worked. And it's not going to work. It's getting worse, as I'll show you in a moment. So we need to abandon OPV. And this idea that NOPV2 is better and more stable, that's just wrong. If you look at the data critically, you'll see that it's wrong. Okay, so this is data. This is a Lancet paper directly refuting some of the things that were written in that article in the GPEI website, this thing here, which you should never have any faith in an article like this. This is basically a blog post, and there's no references. You should take it with many grains of salt. Okay, next. Anyway, this is a summary of the Lancet study. No difference in the effectiveness. So NOPV2, OPV2, both effective, poorly effective, right? Look at the numbers, 1%, minus 23%. It's really bad. Okay. Now, separately, the eradication campaign is checked by what's called an independent monitoring board. And this is their 22nd report, which was issued in 2023. Here's the link for it. You can download it. It's a PDF. You can read it. And it is here. The Independent Monitoring Board provides an independent assessment of the progress being made by the Global Polio Eradication Initiative in the detection and interruption of polio transmission globally. Okay? So this is a third party who looks and sees how they're doing, which is totally needed. And this is the most honest criticism of the campaign you're going to get, because the campaign doesn't criticize itself. All they do is say, yeah, we haven't reached our goals, but we're going to try harder. You know, it's not about trying. It's about looking at the frickin' data and seeing what's happening. Folks, it's not a football game. I once heard the head of the eradication effort at a meeting say, yeah, we're missing our goals, but we're going to double our efforts. It's not sports, guy. You can't just push people. You got to change your strategy and use the right vaccine. So, excuse me, what does this report say? Okay, Independent Monitoring Board. Sorry, it should be B. Conclusions. All right, so on page 37, there have been more than 41 outbreaks of poliomyelitis associated with the distribution of 700 million doses of NOPV2. So this is NOPV2, this engineered type 2 polio vaccine, which was supposed to be more stable. So in other words, 41 outbreaks of paralytic disease caused by the vaccine. It was supposed to be perfect. It wasn't supposed to cause any outbreaks. It caused 41. That's one case per 17 million doses. Now, what does OPV, monovalent OPV2 do? 1 in 1.5 million. Tenfold better. Yeah, but it's still paralyzing kids. NOPV2 is not safe. Look at what they said in this bloody GPEI thing. It is safe and effective. I'm sorry. One child paralyzed would not be safe. And this is 41 outbreaks caused by this vaccine. So you are either lying, which I doubt, or you've just never looked at the data, the Independent Monitoring Board. Or if you have looked at the data, as Albert Sabin once said to me, you're choosing to ignore it. Oh, I wish Albert Sabin were around. Oh, NOPV2 is not safe. It is not allowed to be used in the U.S. OPV is delicensed because it paralyzes kids. And NOPV2 is a derivative of OPV. It's not licensed in the U.S. either, and it's never going to be, because we don't use vaccines anymore that paralyzes kids, thank goodness. And why should they use them in Africa? So NOPV2 is used whenever there's a type 2 outbreak, like Gaza or in many other countries. The rest of the world either uses IPV like the US or bivalent OPV types 1 and 3. And so most immunization occurs with a vaccine that does not have type 2 component. And so there's a growing type 2 immunity gap. And that's why we have such big outbreaks of type 2 polio. It doesn't help to go in and just stop the outbreak or try to. You need to give people immunity to type 2. Okay, another conclusion from the Independent Monitoring Board, page 39. 2 full vaccination rounds plus a mop-up have been successful in stopping breakthrough transmission in 20 of 28 countries to date, a success rate of 70%. When 3 out of every 10 outbreaks are not being contained, it's a clear sign that the current approach is not yet sufficient. As I said, these vaccines, OPVs, do not uniformly stop transmission. They can, but they don't always, and that's why we still have problems. And we need another approach, which is not just hammering with NOPV, giving NOPV over and over again. And page 54 of the Independent Monitoring Board. This was 2023. The first strategic goal is to interrupt wild poliovirus transmission globally. This is done, again, using bivalent OPV. So we have types 1 and type 1 primarily circulating. And we use bivalent OPV, types 1 and 3, to try and control that. The goal was missed. We did not interrupt wild poliovirus transmission by the end of 2023. The goal was missed. Endemic polio transmission still occurs in Pakistan and Afghanistan. In fact, the virus lineages in Afghanistan have expanded and moved into Pakistan. So these viruses, these type 1 viruses, are spreading as well. So OPV does not interrupt transmission, as evidenced by the continuing transmission of type 1 poliovirus in Afghanistan and Pakistan in the face of campaigns using bivalent OPV. By the way, the Taliban has interrupted all vaccination against poliovirus in Afghanistan and Pakistan. And so the situation is going to get even worse. There's going to be even more transmission. Okay, so a few thoughts just to end this, and then we can talk about it. So what is this NOPV2? Well, it was commissioned by the Gates Foundation and WHO because they were using lots and lots of OPV, and it was paralyzing kids. And they said, this is unethical. Can we make a vaccine that doesn't paralyze kids? And so that was the impetus for making NOPV2. And the U.S. stopped using OPV in 2000 because it was unethical to paralyze children with the polio vaccine. But I just showed you the data. NOPV2 also paralyzes kids, albeit at a lower frequency, but it still does. And I don't want a single child paralyzed by the vaccine when we have a vaccine that does not paralyze kids. So if use of OPV is unethical, so is that of NOPV2, because NOPV2 is a derivative of OPV2. Now, the Gates paid for the development of NOPV2 billions of dollars. They pushed very hard for its approval because they claimed its use would stop type 2 vaccine-derived outbreaks within 6 months. Now, there are no wet virologists at the Gates Foundation. They're all modelers. So they based this conclusion on their modeling, which is wrong, because the use of NOPV2 has not stopped type 2 vaccine-derived outbreaks, and they show signs of getting worse. They're increasing in number. All right, one more. So here are the human-caused cases of poliomyelitis. Over 2,500 cases of paralysis in the 6 years after the removal of type 2 OPV from OPV. Over 2,500 cases. Look all over the place, many of them in Africa, where immunization rates drop. We go in with OPV2 and we cause cases of paralysis, and the virus continues to circulate. So many, many thousands in Africa. Peru in 2023, 6 cases of type 1 derived poliomyelitis. Angola, type 2. New York, 2022, we had a case of circulating vaccine-derived polio. 2 cases in London. Well, they weren't paralytic. In London and Budapest, these were viruses found in the sewage in the absence of poliomyelitis, but showing that the virus is replicating in people. These were type 2, vaccine-derived type 2. Ukraine, circulating vaccine-derived type 2. Gaza, as you know. Jerusalem, Egypt. India has had cases of circulating vaccine-derived polio, 2 cases. Indonesia. And then the type 1 circulation, Afghanistan and Pakistan, and it moving outside of the endemic zone, the traditional endemic zone of those countries in 2024. So all of this in the face of massive immunization efforts with bivalent OPV or NOPV2, and the numbers just keep going up. You need to change something. It's not working. The vaccine is not safe. It's causing polio more than wild polio. And we have an alternative, folks. We have IPV that works, that doesn't cause polio, and we need to switch to it now. And people say, oh, it's injected. This is going to be a problem. This is nonsense. You give it as part of routine immunization as a pentavalent formulation with DTaP and other immunogens can be done. The use of OPV not only causes paralysis, it reseeds the environment with poliovirus, which circulates and causes paralysis. So that's why I think the Gaza immunization is wrong, because they're reintroducing type 2 poliovirus, which is going to circulate in all the individuals. Even if they're vaccinated, they're going to still get infected because the vaccines don't prevent gut infection. It's another thing that people assume is the case, but it's not. So the eradication effort is woefully misguided. It's run by bureaucrats who don't want to understand the underlying science. They think they can take a vial of vaccine off the shelf and put it into kids, and that solves the problem. It's not the way to do it, folks. And so I thought it was very important to hammer this home to this audience, with which this issue would resonate, I think. Okay. Let's see if we have any related questions related to. Then I'll go back and answer other things that you've asked as well. Let's see. What was the last one? Yeah, well, Mark says OPVs were phased out in Australia years ago. Many countries are using IPV because it's the right vaccine. You'll have to always immunize with IPV because it will not eliminate virus circulation, but you won't have paralysis. And really, that's the goal of a vaccine, to eliminate severe disease, not to eliminate virus circulation. The question is, who are the scientific experts pushing NOPV2 to WHO over Salk? Well, basically WHO commissioned Gates to do NOPV2. So there's a team of scientists who created NOPV2. And they have been on TWIV. And we discussed that NOPV2 is not the solution. And at least one of the developers agrees. But, we're talking about a big bureaucracy here that has a lot of momentum. And it's very difficult to change. And so they have to start listening to some other people. I agree. All right. So here's another issue that's really a non-issue. Unfortunately, OPV requires less logistics. That's not a good reason to paralyze kids, especially if you're going to give them routine immunizations. 5 vaccines in one, and one of them is IPV. Sorry, it's not less logistics. I would say IPV as a hexavalent vaccine is less logistics than that, because you're giving many vaccines at the same time. Speaking of Gaza, I heard a conversation. I wonder if this affects polio vaccines. Well, the vaccines have gotten in. It's gotten in. So they've done a good job of immunizing people. So if you're immunized with either vaccine, you shed polio virus in feces, always. Yeah, I had a picture, and it didn't make it in the update. Sorry. Mike says, Gaza's unsanitary conditions. What specifically could this mean? And what are the protections we can agree need to be met for citizens? Well, when you have sewage open and people contacting it, then people can get infected from sewage. Now, typically one doesn't get poliomyelitis from sewage. You get it from another person. The sewage is just a sentinel for the presence of virus in feces, right? So certainly in a war-disrupted zone, people can get contaminated. So drinking water can be contaminated with sewage, and it would have polio in it. So that could be one venue. So you need to make sure the sewage is sewage and kept separate from people. I think that's the bottom line. Yes, as Amy Rosenfeld says. Hello, Amy. The excuse of needles for IPV is no longer appropriate. Hexavalent, pentavalent vaccines combine IPV with others. That's absolutely right. And yes, all polio isolates are shed in feces, whether you get one vaccine or the other. with such low effectiveness, how come that OPV2 got given the go-ahead? Well, that was one country, one dose in Nigeria, okay? But, the effectiveness varies from country to country for reasons that are not clear. And so that was not the basis for the licensure or the emergency licensure of NOPV2. But it just shows that it's not the global choice for taking care of polio, for sure. Let's see. Amy says too, like the definition of unsanitary is not specific to polio. Before polio was detected, it was clear that waterborne diseases, including hepatitis A, dysentery, etc., transmission was increasing. That's absolutely right. Yeah. She said, I watched a live stream from a hospital in Gaza, could not figure out how they knew how many drops were given to each child. They had these little bottles, and some may have gotten a drop, some moved. Yeah, it's not the greatest condition, but so yeah, there are uncertainties. Whereas putting a needle in an arm is pretty certain, right? Why the hell are they not looking at the literature? What? You want them to read? Good question. That's the point I'm trying to make here. It's all there. The numbers are all there. And it's in the mainstream media. I mean, the New York Times recently published an article about the switch, the disaster that the switch was, and misrepresented a number of things because they get fed stuff by spokespersons, which are the reporters get fed stuff by spokespersons, which isn't correct. So, in fact, I wrote a letter to the author, and I put one of the videos I put on is basically that letter response to the inaccuracies in that column. Is there any hope for an inactivated vaccine for the gut? Vista, we can't, after 2025, we can't do any more polio research. So this is another pet peeve. There's a thing called polio containment, whereas at the end of June 2025, you can't do any more polio research. You got to destroy your stocks. Why? Because they think the lab is a source of polio. Are you serious? 2,500 cases. That's times 100 people infected. There's way more poliovirus in the environment than in any laboratory. The idea that you have to destroy polio stocks is just absurd. And, it's not because I want to work on polio. I don't have a lab anymore. It's just we need to do polio research to answer questions like this. Can we make a better gut vaccine? Something that doesn't have to be injected. You can't do any research. It's dumb because they think OPV is the answer, and it's not. Folks, you need to write your congressman. Yes. So Chumakov, one of the authors of NOPV2, wrote a science article with Stanley Plotkin, and they said basically NOPV2 is we shouldn't use it. It's unethical to use it, and we should use IPV. Any other attenuated vaccines causing secondary infections? I don't know about secondary infections, but I don't know of any other attenuated vaccine that causes the disease it was designed to prevent. As far as I know, polio, OPV is the only one that does that. Have you considered sending an opinion article to the Times? Yes, I have considered it. I chose to make a couple of videos. I think it's a different audience. I might still do the Times. They don't have to publish it, but that shouldn't be stopping me. I agree. I really should do it to summarize all of these issues that I've talked about. Okay. go ahead and spread the info. I'm going to release this video as a standalone so you can look at that too and give it to people. Can tonsillectomy and adenoidectomy reduce IgA? I don't know. It's a good question. On the last clinical update, Daniel was saying there are a lot of people that don't have IgA. They don't seem to suffer more respiratory mucosal infections than other people. I think it could reduce them, but then other tissues could step in. I don't know. I don't know the answer, Patricia. It's a good question. Who's responsible for the decision to destroy stocks of polio? The CDC. I think, well, look, I'm going to say wrong things, but first of all, it's voluntary. You don't have to. But if the CDC recommends it, your institution is likely to tell you to destroy it. And so, for example, if your institution, if you're working on polio, they're going to say you can't work on this. The CDC says you can't, and you could say, well, it's voluntary, and they say you want to work here, it's not voluntary. So that's how it works. Could I give a mini lecture on viral infections and fungi? Yeah, that would be cool. That would be cool. We could get Arturo on here, right? That would be fun. Does polio have a non-human host in nature? No. Humans are the only host in nature. Unbelievable that research is going to be stopped. That needs to be heard far and wide. So is this news to most people that polio research is going to be stopped next year? I thought most people knew that. But again, the awareness of polio in the U.S., I think, is very low because people have other issues and they figure polio is done, right? World Health Assembly made the decision that was authored by Phil Miner and Mark Palanch, the decision to destroy stocks. The labs are not a threat because there's way more poliovirus in the environment. Exactly how much we don't know because we don't look for it. And so, the fact is that 1 in 100 polio infections are paralytic. So if you have 2,500 cases of paralysis, there are 100 times more infected people, minimum, in those areas. So it's a lot of polio in the environment. Okay, so that is a human-made polio, series of polio outbreaks, which maybe other people didn't know. You didn't know that most of the polio globally was caused by the vaccine, did you? And in that case, we shouldn't be using that vaccine, should we? All right, let me go back and grab some of the questions that I have skipped over. Of course, you can keep asking questions about polio or other things as well. I just want to go back and make sure we cover them. Okay, so Kang wants to know if there are viruses resistant to interferon. And the short answer is yes. The long answer is it's hard to really know in the natural host. You can take cells in culture and infect them with viruses and the presence or absence of interferon and make conclusions about interferon sensitivity. I'm not sure what that means. What you'd really like to know is in a host, does an interferon response measurably impact the course of disease? And you could do it in an animal model, right? But then you don't know the relevance to humans. Yeah, there's a lot of uncertainty in science. That's the way it goes. There's no simple answer. So yes, you can identify viruses that are less sensitive to interferon in animal models, for sure. And many of those encode antagonists of interferon in their genome. And then there are viruses that are exquisitely sensitive. But the key is in people, and that's where with COVID, we have a lot of data showing that one of the main risk factors for severe COVID is not having a good interferon response. So apparently that makes a big difference early in infection for SARS-CoV-2. And so people who make antibodies to their own interferon, they are more at risk for having severe COVID than people who do not. So that kind of data can be used to tell you that, yeah, SARS-CoV-2 is quite sensitive to interferon. And if you make a good amount early on, you can control the infection, severity of the disease. So a pig snake bear asks, I'm looking for challenge experiments with bird flu that don't involve injections. I can't find any. Do they ever do them like that? Well, yes. You can affect a chicken and then put it in a cage with another chicken, right? And then you are looking at chicken-to-chicken transmission. Those certainly have been done, but I don't, offhand, I don't know. I can't point you to them. It's been done for many other viruses as well. The original chicken is not injected, but they have to have virus put in them. Otherwise, it's not really a challenge experiment. But you go in the literature, you find it. You just need to know what to look for. You need to know how to use PubMed. And everyone says, oh, yeah, I go to PubMed, I find the answers, blah, blah, blah. Sure. It's not easy because there's a lot of chaff there. So you have to find the right study that does what you want to do. And it's not easy because you're going to get the abstract and you're not going to get all the information. And after 3 or 4 papers, you're going to throw up your hands and say, I don't have time to do this anymore. It's not easy. You want to go through hundreds and hundreds of papers to find the right answer. That's the challenge of going through the science literature, and why when people say to me, yeah, I read the literature, I make my own decisions, I don't believe them because it's really hard for me to do. Why do I think you would be able to do it without any virology training? Yeah, here's an expansion. Just exposing sick birds to healthy ones. Yeah, of course they do them like that. As I just said, they put an infected bird in a cage with an uninfected bird. They do that for all kinds of virus infections. It's done for COVID too. Transmission models for COVID. Yep. So when's the best time to get your flu shot? So Daniel was asked this question on the last clinical update. He said, there's no hard data that tells us when you should get it. Yeah, some people think late September to October. Well, it's late September. You better get it. But really, there are no data that would inform us as to the best time to get your flu shot. What time of year? Yep. Yep. The flu vaccine, I presume that's what you mean, is a little hit and miss. I thought I had a slight cold. I tested for influenza A. My dad is in the hospital with it. He's 95. Well, it kept you out of the hospital, Mark. And 95 is tough. Hopefully, he gets out of the hospital, Mark. And Michael says, just to expand, some acquaintances tell me of personal experiences where something happened to their relative and blame it to the vaccine. Yes, that's common. This is the issue of uncertainty, right? And that's in medicine, right? It's famous. Medicine is full of uncertainty. I cannot tell you what is going to happen to you with 100% certainty. Here are the odds, and people don't like that. So they want to blame something. This is a blame society, right? So, yeah, I'm going to blame the vaccine without actually looking into it and seeing if the vaccine, in fact, could be at fault. Maybe it's not. But these are hard cases, yeah, for sure. Here's an interesting question. I've heard and read many things about DNA, RNA, tRNA, and mRNA. Can you please briefly talk about the history of exploration of inner nucleic envelope proteins and enzyme-like structures? No, I can't briefly talk about it. Inner nucleic envelope proteins and enzyme-like structures. You're talking about the cell nucleus. You want to know what's on the inside of the envelope, and you want me to tell you the history of it. Well, we can't do that tonight. It would take too long. But as you know, the nuclear envelope surrounds the nucleus. Inside it is the DNA. And the nuclear envelope has pores, which are very complicated structures that regulate the traffic of materials in and out of the nucleus, embedded with other proteins as well. The main function of those is to regulate traffic in and out of the nucleus. And those were discovered many years ago. If I'm not mistaken, Amy, George Palade first involved in discovering those structures. And then over time in the 60s, and then over time, they became elucidated by biochemistry. And now we have a good idea of how they work. And they're very sophisticated machines that regulate traffic back and forth. Condor Kid, do you think the media science correspondents' focus on variants caused people to think vaccines are no use and virus got weaker versus vaccines worked and people did not get as ill? Wait, did you think the variants caused people to think vaccines are no use? Oh, and the virus got weaker versus vaccines worked and people did not get as ill. Yes, I do think so. I think the variant narrative was ridiculous. We fought so hard to say to people, you don't know anything about these variants. And well, I published a video early in the pandemic. UK variant, doesn't matter. I got hate mail from people in the UK. You have to take that down. It's misinformation. No, it's your misinformation. I'm just telling you the facts, which are we don't know anything about these variants. And for you to think that you should be scared of them is absurd. You know, the alpha variant in the UK emerged in December, what was it, 2020? And it was at a time that the UK was reopening. So, do you think that reopening and people coming back together is going to be associated with transmission? Yeah, of course. It may have had nothing to do with the variant. And to this day, there's no evidence that any variant is more or less transmissible or more or less pathogenic than any other variant. Bottom line, there's zero evidence. There's some circumstantial evidence, but there's zero evidence that variants matter at all. I said on the clinical update a few weeks ago, I would love to have a cohort of people. Now I just erased myself. Oh, darn. Here we go. I would like to have a cohort of people who got the original 3 vaccine doses. I know one person who fits that category. And see how often they got infected and what kind of disease, not infected, what kind of severe disease, what kind of disease they got over the years being infected with different variants. And I suspect that in people under 65, if you had those original 3 ancestral SARS-CoV-2 spike, 3 shots, you would be protected against severe disease because the T cells that you made protect you against all the other variants. The issue arises that some of the variants are immune evasive, so your antibodies don't work, and you get an infection. And you feel crappy, and you say, it's that variant, but you didn't get in the hospital because your T cells worked. And of course, if you're over 70-ish, then any infection is going to send you in the hospital and can be severe. So variant is irrelevant, in that situation. Now, what are we doing now? As I told you folks a couple of weeks ago, CDC is telling you to get a booster every 6 months. So you make a bunch of antibodies that are going to reduce viral loads and maybe make you feel better. But that protection is going to go away after a while, and it's not a great public health strategy. I don't think it's necessary, and there's no evidence that it is necessary. However, if you load up your respiratory tract with antibodies, it's going to have some impact. So that's their justification. Suggestion 4, mini measles lecture, Paul Dupre. You know, Paul has been on TWIV a couple times. Pete, go check it out. Yeah, he's got a very strong Northern Irish accent. Absolutely. Do all vaccines with adenovirus vector have a rare risk of thrombosis? Ooh. So we had the vaccines that were originally licensed here in the U.S. by J&J or EU aid, which I think was AD26. And then we had the Chadox vaccines. Let's look. Do all ad vectors cause thrombosis? Yeah, all of them had rare thrombotic events associated with them. Yep. And until that's fixed, I would stay away from them, right? That can be fixed. I'm not sure it will be because we know what causes it. Yeah. So Jill says, Neil deGrasse Tyson occasionally does reaction videos to sci-fi movies. It's hilarious and informative. Also, how do we get the TWIV gang on Star Talk? Neil does a lot of science cross-pollination. I don't know. You know, I just don't know how to get in with any of these people. And yeah, it would be great to have a bunch of the TWIV gang on Star Talk. It would be a lot of fun. He probably has no idea we exist. And if anyone knows people in his circle, please let him know. We'd love to do that. What's your take on psychedelic assisted therapy and mind medicine? I don't know. I have no idea. That's not, that's out of my field. And I'm a virologist. I can tell you some things about viruses, but I don't know anything about this at all, Brian. I'm sorry. Okay, Hans has a question. Viral adjectives such as respiratory or enteric refers to how a virus enters the body, to the type of cells where it replicates in, or to its tropism. What a great question, Hans. Really good. Is rabies a muscular virus? So it can be either, right? So we talk about respiratory infections. These are viruses that infect the respiratory tract. Enteric infections, viruses that infect the enteric tract. But then it would also refer to what kind of cells the virus is reproducing in, right? To tissue tropism. So as I think the key here is that you understood the nuance to phrase the question properly. And so now we can answer it really precisely. So yes, it is the site of disease. It's the tropism. It's the kinds of cells that are infected. Is rabies a muscular virus? Well, rabies is moving up the, it gets into the muscle, but then moves up the nerve. So I would say it's a neurotropic virus. Yeah, rabies is neurotropic, right? John, thank you for your support. Vincent, can't be said enough. You are a role model for what to do with expertise if your basic needs aren't being met, and very few people do it. Nonprofit for the betterment of humanity. May we all follow suit. Thank you, John. I appreciate your support and nice words. I was thinking of this the other day. I always felt I should give back my expertise in some way. I was fortunate to get a great education, right? And I accrued lots of interesting information about viruses. So I started teaching. I wrote a virology textbook. I started blogging, started podcasts. And, today it's coalesced into Microbe TV, giving back what I know. That's what it's about. So thank you. Appreciate your support. If you were ever pushed to do advertising, what services might you be comfortable with? Um, Knitting, gutter cleaning, um, stump grinding, that sort of thing, which has nothing to do with no conflict of interest, which would be interesting. I would be very happy to support to advertise for other nonprofits that have a good cause, but not pharmaceutical, not antivirals or vaccines, nothing, no biologicals, because it's just too close, obviously. Hello, Abdul Aziz from KSA 25C, cooling off there. Yep. Do you have any speculations on whether the Moderna norovirus vaccine? I have no idea. No idea. I don't know what the numbers look like. Let's see. Are they out? The mRNA is not going to be for everything. They have a bunch of vaccine candidates in testing. I don't know anything in particular about the mRNA candidates. So no, I don't have any speculations at all. Speculations are tough, right? Any thoughts on the Nature Medicine article, post-hospitalization? COVID-19 cognitive defects at 1 year are global and associated with elevated brain injury markers and gray matter volume reduction. So the issue of gray matter volume reduction has come up before. And some of those studies were highly criticized because they weren't properly controlled. And, the controls were really inappropriate. And so this is a new contribution. And I think Daniel's going to cover this in the next clinical update. So let's see what he has to say. Yeah, getting some pickup in the mainstream press. Well, these long COVID papers typically do, because there's a lot of demand for understanding what's going on. Echoes in the Dark says, I always tell people, anyone that tells you this issue works like this and this will always be the solution, to get suspicious. Yeah, solutions change always. The world is a fluid place. And Echoes, by the way, is originally from Nicaragua, currently in Maryland. So Echo says in Nicaragua, there are very few cases of polio. The government implements biannual vaccination on top of ready availability at primary care centers. Well, that's good. And what vaccine do they use? Do you know? Hopefully it's IPV. I still love this comment. Why the hell are they not looking at the literature? It's a good one. Why the hell? Good questions tonight, folks. I'm looking for more. This is interesting. I guess the mentality of governments choosing correctly living with COVID switched to living with polio. To a certain extent, I think so. And so for here in the U.S., people don't really care about it, right? But I think we have to have a broader awareness, a global awareness, not just the U.S. We're about the world because we can make things work in the rest of the world if we want to, right? I'm sorry, Andrew, that your grandmother passed. You used to watch the weekly update with her. I'm sorry. My condolences. Lee says most people do not know the polio research will be stopped. Well, we are trying to spread the word about this and all the other faults about eradication. Here's the thing. The more polio there is globally, the more there will be here in the U.S. And we're lucky that vaccination coverage is quite high here in the U.S. with trivalent IPV, types 1, 2, and 3. But the more viruses circulating elsewhere, they're going to come here and circulate. And so if there are any immunity gaps, we're going to have outbreaks of polio here. And at some point, parents are going to stop immunizing their kids against polio, just like they have against measles in many states. And we have outbreaks of measles all over the U.S. Crazy. When people come back to their senses, I don't know if they will, Artemis. I don't know if they will. Can you make new polio stocks from viruses that people shed? You couldn't take the DNA sequence and make new viruses for sure. But, once you've stopped research, it's really hard to go back. And so the bureaucrats running this containment program where you have to get rid of all polio by June 2025, they're like, oh, there's nothing we can do. It's going to happen. And no matter what you say, this is so stupid. You have really good scientific arguments why containment doesn't make any sense. And they'll say, no, we can't change this. It's like this is the corporate momentum and you cannot change this. This is what pisses me off when corporate attitudes infiltrate science. So, yeah, this is our agenda and you can't stop us no matter what you say. They take it personally. No matter what you say to them, you can't change it. Oh, my gosh. Next time Dr. Griffin is on Brian Lehrer show, he should discuss this NOPV. We'll have to brief him, right, to make sure he gets it right. But I agree. I think this is something that should get a broader audience. Did you know there's more polio caused by the vaccines? Did you know we're about to stop all polio research and this is a bad idea? Did you know eradication is not working? Then people write op-eds to the New York Times saying, oh, eradication is wonderful. Look, we're almost there. Clueless. All right. What else here? Symptoms during the second week don't correlate with RNA copy number. That's why it's not a matter of continuing use of Paxlovid. Having said that, what do you make of Fauci's decision a couple of years ago to take more Paxlovid? Bad decision. Very bad. First of all, he's a doctor. He should know better. The prescription is for 1 5-day course. He should have done that. And he's a visible physician. So he's saying, I wonder if virus is replicating in me. He's speculating. And that was just wrong. And I think that led to a lot of the silliness about Paxlovid rebound. Yeah, that was a bad move. I understand he made a mistake, but he should have admitted it. He should have, I shouldn't have done that. It was a bad move for this reason. But he frequently did not admit his errors, right? Now, I don't want to bash him at all, but he could have used a little error correction, right? How do you know if you make good interferons. Unless you're the subject of a research study, you don't. And, if you get lots of infections, it could be a clue that you don't make good interferons. There are some labs that study people who are at risk for severe, say, COVID, and they see, you're making antibodies to your interferon. So you could enroll in a study like that, but it's not something that you're going to get off the shelf. That's for sure. Serge is my fan from Ottawa. I like to watch your virology courses when at the gym. All right, pumping iron. You got it. You and Angela Mingarelli. Why don't they vaccinate large bird farms for bird flu in the USA? It's all right. So this is what I've been told. It's very expensive to do this, and I'm not clear how effective it would be because there are lots of wild birds that have H5N1 avian influenza virus. So I suppose it would protect the flocks, but wouldn't impact the virus spreading via those other birds, right? We had a discussion about this on a TWIV, and that was what Alan came up with. It's just not cost effective. Was the clinical infectious diseases retrospective on lower mortality with REM plus Dex versus Dex alone covered with Dr. Griffin? Recommend changing NIH treatment guidelines. Gee, I don't know, John. I can't remember. They all blend after a while. I can check. I'm going to make a note. It's an interesting question. You could ask him. You could send an email. I know you do sometimes, and then tomorrow is the day. We can ask him that. Julie, I'll be flying within the next 2 weeks. I'm over 65. Should I wear a mask? Amy says that they don't vaccinate birds on farms because the lifespan of the bird is too short. Thank you, Amy. If you don't want to get infected, yeah, you should wear a mask. A plane is a great place to get infected. Now, everybody I hear, I went on a plane flight and I got COVID. Yep, there's COVID on them planes. And Daniel talked about that in a recent clinical update. How yes, despite the heap of filtration on the planes, you can still get SARS-CoV-2 infection. So it's up to you. If you wear a mask, you're going to enhance your protection against getting infected. I prefer not to be infected. I'm 71, so I would wear a mask. I wear a mask on the train. Yeah, a side effect of the flu vaccine is that if you get ill afterwards, you'll blame the vaccine and tell everyone about it every chance you get. Yes, exactly right, Condor Kid. This is one of these social phenomenon, right? Yeah, you're going to associate it with the vaccine or anything untoward that happens to you after you get the vaccine. You're going to blame it on the vaccine. This is a social phenomena, right? It takes a lot of training not to do that. It takes many office hours of listening before you will say, oh, I got the vaccine and I tripped, but I know that they're not related. A mask worn properly diminishes the chances of catching any airborne disease. There's a new review article out, which is getting some press, which underscores that a properly worn mask has a measurable impact on reducing respiratory infections. I'm going to cover that on TWIV Friday. Silent Runner got the 3 original shots. I have not been sick at all. I'm 70. Great. We have now N equals 2. We have to assemble this cohort of individuals who just got the original 3 and look at how they fared versus people who got all kinds of other boosters. And that would be such a cool study. I would love to do that. No, I'm not doing it. Mark, what were the state-of-the-art questions polio researchers were investigating before research was banned? Oh, so polio is an incredible model virus for so many fundamental processes, so many questions about nucleic acid reproduction, protein synthesis, changes to the cell, changes in membrane functions in the cell, membrane trafficking, protein trafficking, um, all sorts of macromolecular processes, nuclear export and import. All of these are impacted by poliovirus infection. And many people were using the virus to study them. And, if you look at the history of what was learned with polio, it's huge. Amy and I are writing a chapter for a textbook on coronaviruses, and it's just remarkable how much we know from studying just polio. Now, I'm not saying we need to restore polio research just for that reason. We need to keep doing polio research because we don't have a vaccine that's going to fulfill what we need, and we need to do more research. Right? OPV is not the answer. IPV is good, but you have to use it forever. So if you want something else, we need to do some research. But oh man, so many things were discovered on using poliovirus. Really amazing. Mark is a novid. Yep. Novid. No COVID. Aerosone. Are there any studies looking at possible effects of frequent repeat vaccinations? I don't know of any systematic study, right? Because, well, you're not supposed to get repeat frequent vaccinations. Only you're supposed to get what's recommended. So I'm not aware of any. The only one I know of is the German fellow who got 260 some odd COVID vaccines, within the last couple of years. And he seems to be okay. But that's an N equals 1. So I don't know the answer. It may be that it's innocuous, but we just don't know. Here, alpha variant in the UK, Will. Prime Minister talked about it early in December. What he did not say was that studies of samples found the first occurrence dated back to September 20th. Yeah. Well, he Boris told everybody to go play, go back to life. So they all went back to their games and their pubs, and they spread viruses. And the alpha variant happened to be there, and it spread. Right place, right time. Is there a vaccine being developed for urinary tract infections? Don't know. As Daniel Griffin says, over 90% resolve without further treatment. Let's look. I would be surprised if there weren't vaccines being developed. Well, there is a polyvalent bacterial vaccine developed for UTI. It's available in 26 countries, called MV140. That's all I know. Many bacteria can cause UTI, so I don't know how that would work. It reduces recurrent urinary tract infection. This is a paper. In Canadian Urological Association Journal. So check it out. Stub grinding. Yes. Well, that's a reference to an old TWIV. If you're an old TWIV listener, you'll get it. What are the current symptoms of COVID? They're the same as they've always been, right? There's a lot of potential, but you've got your cough, sore throat, fever, runny nose, and then the more severe, low oxygen saturation, lung problems, typical respiratory tract infection. Knitting, that's another thing I would do advertisements for. That's pretty innocuous. Yeah. Mark, thank you for your support of science communication. Mark has been with us for many years, an early supporter back when we used to do advertising. Mark was one of our advertisers. We advertised a computer storage system. Actually, that's an innocuous advertisement that I would take. We used to do ads, and the storage system that Mark worked for, we advertised them, and then we did Curiosity Stream, which is a streaming sciencey video service, right? And Blue Apron. Years ago, we were one of the first people to do Blue Apron, and I said, nobody's going to want this. And all these scientists wrote in and said, oh my gosh, this is great. I don't have time to cook, and this solves my problem. Wow, I didn't know. So thank you, Mark, for your support. I can promote fashionable eyeglasses. Yeah, it's true. Today's eyeglass. Yeah, I like lasagna very much. And you know what I like is the new sheets where you don't have to boil them first. You just lay them in the pan with layers of sauce and whatever, cheese, meat, whatever you want to put in, and then you bake it, and it cooks while it bakes. That's great because the boiling and picking them out of the water, and that was a pain. And I'll tell you what I did the other day. I love eggplant parm, but gosh, I hate slicing it up and, squeezing the water out and then breading it and frying. Blah. So I went to an Italian emporium. They sold sliced and breaded eggplant. So you just bring it home and you make your eggplant parm and you bake it. Oh man, it just reduces. So I could advertise food. Yeah, I could advertise food too. That would be fun. But I don't want to advertise the usual stuff. Everybody advertises Squarespace. Oh my God, it's so boring. How many people need Squarespace? Yes, anyway. So Vanity, her symptoms were extreme fatigue, pink eye, and a cough. There you go. I saw the announcement of flu mist, FDA approval. Flu mist is the same as any other flu shot. It's no better. Yep. Thanks, Visto. Glad you liked it. It clarified some of my fuzzy understanding too to make a lecture. It always helps. It's interesting. If a heating pad is used to raise the temperature of virus-infected tissue in the nasopharyngeal area or lungs, for example, is there a risk of resistance developing similar to antibiotic resistance? No, I don't believe that happened. Well, no one's investigated it substantially, but I think the evidence that raised temperature is antiviral is kind of thin, so I'm not sure that this would be an issue. But you have to be careful when you're heating up body parts like that you don't hurt yourself, right? Margot was able to educate a patient asking if she should get the RSV vaccine in a retirement facility because of all I learned here. Well, that's lovely, Margot. Thank you so much. I'm glad that you have learned. That's the key, right? That's the goal. We want you to learn things. They should have polio wastewater surveillance. Totally, the U.S. should. Much of the world should. But the U.S., the CDC refuses to look for polio in wastewater. You should object. You should all write en masse to CDC. Bombard them with paper letters. We want wastewater surveillance for polio. You do it for SARS-CoV-2, why not polio? Yeah. Remind me, how long does T-cell memory stay viable for a middle-aged woman? It depends on the pathogen, right? And The longest ones that I know of, SARS-1, 17 years of T cell memory in some of the original patients. But, that's one, and we don't know. It's just not well studied. So, if you get infected, excuse me, the key is to get infected at a younger age, less than 65, where you can generate good T cell memory. And then how long they last, we don't know. That's what you're asking. I understand that. But really, people haven't studied it, and that needs to be done now. But it's going to range probably from years to maybe decades, like SARS-1. Thanks, Steve, for your contribution to science communication. Really appreciate it. Everybody is very supportive tonight. I really I'm grateful for that. Jeff says, I didn't know polio research would be stopped. That's one of the stupidest decisions that could be made. Since I missed that, who made that decision? Well, it was developed by WHO in consultation with Phil Miner and Mark Palanche. These are Mark used to be at the CDC, and Phil used to be at the NIBSC in the UK, the kind of FDA equivalent over there. They architected this polio research cessation. And yes, it is not a good decision. Could perfect IPV cover over time exclude poliovirus from a country? No, it doesn't. It doesn't stop gut replication. It's very clear. Even a well-immunized population with IPV, the virus can still infect the gut, so you would not eliminate it, no. Did the decision makers think injections were not practical and the drops were better than nothing? Well, the decision makers did think injections were not practical, and they also thought that the drops were great. They thought OPV was perfect, that it was safe, and it stopped outbreaks, and it stopped transmission. And none of that is correct. None of it is correct. So you would think that the experts, the decision makers, would know better, but they didn't read the literature. Kang writes, is the CDC, by banning polio, giving the impression that there are lab leaks? They would like to prevent lab leaks, right? They say in an post-eradication era, the only source of polio is a lab. So let's get rid of it. The problem is we're not in a post-eradication era. We're not even close. The waterways are full of poliovirus. You could just see all these cases we have all over the world, right? So it makes no sense to destroy lab stocks at this time because labs are not a threat. But yes, they're saying in a world where we have no more poliovirus, the labs would be a threat. But there's never going to be a world with no polio. It is totally impossible that we would eradicate poliovirus from the planet. It's circulating silently everywhere, and we're just not looking for it. That's why you don't hear about it. But, when we had that case of polio in Rockland County in New York in 2022, CDC immediately went and did wastewater surveillance in the area, and they found poliovirus gangbusters. Wherever they looked, it was in the wastewater in the New York area. So if they looked in Chicago or Minneapolis or who knows where in the U.S., they'd find it. But they're not. The fact that Robert Redford doesn't look is unforgivable. Why? Why is he different from anyone else? He chooses how to spend his time. He doesn't want to look through the literature. Now, if I weren't a scientist and I had lots of money and I wanted to investigate a science question, I would hire a smart PhD student and say, I want you to go through PubMed and we're going to find papers on this topic, and I want you to print out the whole paper of each one and find, or you don't have to print it out at first. You can just download the PDF and find all the data with respect to this, and we'll meet every week and we can go over the data, right? And you pay them hourly to do. If they pay them $30 an hour to do that, you'll get your work done. I don't know if you get an answer, but you get your work done. About 2 weeks ago, you criticized the use of viral load. Daniel presented a study with the term. Which is it? Why use viral load in HIV but not SARS? But because we can clear SARS. It's not viral load, right? It's viral RNA copy number. And yeah, they got away with it for HIV because we weren't around to call them out. But it's not correct for HIV either. For HIV, it's harder to do infectivity than it is for SARS-CoV-2, but it's not an excuse. You should just call it viral. For many studies of HIV, they do call it DNA copy number, for example, for RT-PCR. That's the objection is viral load means infectious viruses. So if you're doing PCR, it's not measuring infectious virus. So I want you to say viral DNA copy number. That would be fine. And for many viruses, they do that. Like I saw a CMV study, cytomegalovirus, where they were measuring viral DNA in the blood, and they called it viral DNA-emia, right? Not viremia, because it's not virus, but it's DNA-emia. That's perfect. Just define exactly what you mean, yeah? Oh, and KSA, 3 IPV and 4 OPV. That's very interesting. But the OPV are just making things worse. They're just seeding the environment. There's no need for OPV after IPV. So I would advise KSA not to do that should they want my advice, but I'm sure they don't because I'm just a science professor. What do I know? Angry Penguin, many years ago, WHO visited our site. We made IPV. At that time, they said they were targeting 2012. Yeah. They started the eradication in 1988. They started first 2000, then 2005, then 2012, then every year it gets pushed forward. It's not working. Change. Bill says, my brother-in-law is paralyzed by polio back in 59. I'm all for continued research. Amy says they do not vaccinate birds on farms because the lifespan of the bird is too short. Margot says, I must make good interference. I don't believe I've had COVID. I had antibodies before vaccine, got to 3, was tested twice a week. Well, good. It's good. Dr. Susan Oliver uses the terms bollocks. Dr. Racaniello uses the term pisses me off. What? I do? Really? Pisses me off. I didn't know that. I had no idea. Well, I hope it doesn't piss you off. It generally is, Vanity wants to know, is the measles vaccine the worst vaccine not to get? No, I think it's among the worst because if you get measles, it'll erase your immune memory, but you can get it back. But of course, there are other bad side effects of measles, right? Encephalitis, pneumonia. I think the polio vaccine is a bad vaccine not to get because you get paralyzed. You won't be able to walk. And people are complacent about it because there's very little polio, myelitis in the U.S. But yeah, it's a good question. I wonder if kids will grow up and resent their parents getting them sick. I hope they do because their parents are supposed to take care of them. They're being idiots for not vaccinating them. There's zero reason not to vaccinate your kid unless you have a medical exemption, right? So vaccinate your kids. Take care of them. Do the thing you're supposed to do as a parent. COVID transmission is most likely to occur during boarding. Yeah, it's less likely during, unless when people walk up and down the aisle coughing, that could do it. People do that all the time. Why do they cough? And sitting next to you, if you got a cougher next to you, right? Or in front of you. But yeah, I agree that during boarding is the worst. You should certainly wear a mask then. Then when they crank up the air, if you got a clear cabin. Areozone, there's a quote from philosophy historian Yuval Noah Harari, because the truth is a costly and rare kind of information. It's usually easier to connect people with fantasy and fiction. That's great. I love this. Very nice. Very nice. How is IPV made? Yeah, poliovirus is used You have to grow large quantities of infectious poliovirus and then inactivate it with formalin. And so it would be nice to make a vaccine that didn't require that, but we can't do any more research. So it's not happening. Okay, let's see. We're 10 o'clock. And let's see if there's any urgent, a lot of masking in airports. And Vanity, of course, knows all about that, having worked on planes for many years. So take her advice. Are there some viruses that mRNA vaccines excel in providing immunity? The only one that it works so far is SARS-CoV-2. The others are being tested. Who knows what the outcomes will be, right? We'll see if SARS-CoV-2 was a fluke, a lucky fluke for humanity, right? Okay, I think it's time to wrap this up. You guys had a nice little... Oh, look, Sharon is still here, still resonating. Thank you. I'm glad you enjoy it. Lots of great questions, but it is time to wrap this up. Bring your questions back next week. I will be back at office hours next week. What's the date? It is... Wait, is today the 25th night? No. Yeah, next week, 2nd. October 2nd will be our next live stream here at 8 p.m. Eastern. I want to thank all of our mods tonight for hanging out. We had Andrew here tonight. We had Vanity Nutrition. We had Les. We had Andrew, I already thanked. Who else was here? Frank was here early on. One more I'm missing. Les was here. And from Oregon, I can't find his name. You know who I mean. Thank you all for moderating. We appreciate it. And thank all 160 of you for coming by tonight. Great questions, as usual. Great community. Keep it up. You show up here and ask good questions, we get the ball rolling. So keep doing that. So we'll be back next week, folks. In the meantime, I want you to stay safe. Good night, everybody.